Welcome! On today's lightboard, I want to illustrate the most current view on how fish jaws originated. While many different types of jawless fishes once inhabited the planet, a few lineages of which have survived to the present day, such as lampreys and hagfishes, there's no question that the improved biting ability that hinged jaws afford revolutionized the subsequent evolutionary history of fishes by opening up predatory and suction-feeding niches that were previously unavailable. So, how did fishes evolve this important and innovative structure? Let's start by illustrating what we think the cartilaginous skeleton of early jawless fishes looked like. The drawings that I'm going to show here may seem different from what you might see in many textbooks. Those typically illustrate a scenario based on observations of the morphology of developing gnathostome embryos made in the middle part of the 20th century. The reconstruction that I'm going to draw here is based upon much more recent, exquisitely detailed comparisons of development and gene expression patterns in living lampreys, hagfishes, and jawed fishes made by the Japanese scientist Shigeru Kuratani and his colleagues. Observations of fossils of jawless fishes that lived hundreds of millions of years ago also inform this scenario, though of course we can no longer study gene expression and development directly in those organisms. And before I start, I'd like to give a fast shout out to my colleagues Corey Evans and Peter Konstantinidis, who called my attention to Dr. Kuratani's new work on this classic topic in vertebrate evolution. Now to orient us, dorsal is to the top of the screen, anterior is to the left, posterior is to the right, and ventral to the bottom. And I'll start by drawing the cartilaginous neurocranium of a hypothetical ancestral jawless fish here in blue. So I've drawn the neurocranium, and I've also noted the three major sensory capsules, nasal, optic, and otic. And I've added the notochord running posteriorly from the back of the neurocranium. The notochord provides structure to the rest of the body and also runs parallel to the nerves of the spinal cord. During the course of development, the vertebrae form around the notochord. And all of this is quite similar to a modern fish's anatomy. I'm also going to draw the outline of the rest of the fish's head and body, which allows me to indicate where the fish's mouth would be. Now, keep in mind that jawless doesn't mean mouthless. There was still a hole here for the animal to use in slurping up detritus, plankton, small worms, or whatever else it ate. Now, ventral to the neurocranium, I'm going to add several visceral arches, or in other words, the branchiocranium, also called the splanchnicranium or visceracranium. These cartilaginous arches provided skeletal structure to the ventral portion of the fish's head, including the mouth, pharynx, and gills. Now, there appear to have been at least 7 of these arches, arranged into 3 developmental domains defined by the expression of a series of homeobox, or Hox genes. I've color-coded the anterior arch as green, the middle arch as purple, and the posterior series of arches as yellow, with the colors matching the 3 developmental domains. This color coding matches several other of these lightboard videos, and will allow you to identify which parts of the modern fish correspond to the skeletal elements in the ancestor. Now, we can see that in this jawless ancestral fish, these visceral arches had already differentiated in morphology and function. The most anterior seems to have provided structure to the animal's lower lip and the region of the head immediately posterior to the mouth. I'm going to label this as the mandibular arch in green. In the modern lamprey, the cells of this region develop into the lower lip and the velum. The velum is a fascinating structure that moves water through the head of the ammoset larva during filter feeding, then becomes a valve in the adult pharynx that allows them to maintain separate the flows of water over the gills and their blood meal down the gullet when they are attached to a host. Thus, this first arch seems to have been associated with the ventral and posterior parts of the mouth even before the origin of the jaws. Now, the most posterior series of these arches in yellow clearly provided structural support to the gills. We'll call these the branchial arches. So those are the branchial arches, just like they are in a modern fish, where they have the same function. It does seem, however, that the structure of these arches may have originally been simpler than they are today. I'm drawing them here as simple crescents with a few posterior-facing projections. Lastly, the middle developmental region of our ancestral fish contained a single arch called the hyoid arch. The function of this arch in the ancestral fish isn't entirely clear, though it did clearly provide some structural support to the pharynx, or throat of the fish. There might also have been a respiratory function, and certainly there were openings to the gill chamber, anterior and posterior to this arch, as well as between the branchial arches. So these are holes on the side of the fish's body that allowed water to enter and exit the gill chamber, thus supplying oxygen and allowing the fish to respire. I've indicated these with dotted ovals to show where the gill slits probably were in our ancestral fish, and you can still see such openings in modern jawed and jawless fishes alike. So at this point, we can see that our ancestral jawless fish had 3 differentiated developmental regions in the branchiocranium, each containing at least one visceral arch. The anterior one is the mandibular arch, the middle one is the hyoid arch, and the posterior ones are the branchial arches. To complete the drawing, I'm going to add a blue domain here, near the upper lip of our ancestral fish. This is not a visceral arch, but it appears to have been an additional developmental region with a separate set of genetic coding instructions from those that pattern the visceral arches. We'll track the transformation of this region, called the premandibular region, in my next drawing. Okay, so that's our hypothetical ancestral jawless fish. It had a relatively simple body plan, but let me stress that it worked perfectly well for the detritivorous, planktivorous, or micropredatory niches that such fishes inhabited. At the same time, it is clear that this fish was not a major predator, or at least not as efficient a predator as jawed fishes would later become. Next to this drawing, let me construct a similar diagram showing what we think the cartilaginous internal skeleton of early jawed fishes, such as placoderms, looked like. In drawing this, I'm going to keep the color coding consistent so that we can see how elements of the ancestral jawless skull transform into the derived morphology of the jawed skull. I'll initially restrict this to just the cartilaginous skeleton, because while bone actually evolved earlier than jaws did, it was originally restricted to a protective shield around the outside of the head and was not associated with the visceral arches. While bone later has a major role to play in the diversification of jaws, the story of jaw origins is really all about cartilages. So, first let me indicate the neurocranium and notochord of our jawed fish. These elements are essentially untransformed relative to the ancestral state, and so should be familiar. I'll also now add a line showing the perimeter of the fish's head and the location of the mouth. Next, I'm going to add the branchial arches in yellow. The biggest change here involves the dorsoventral patterning. You can see that there are now separate dorsal and ventral portions of these arches. That separation into upper and lower halves appears to have been driven by duplications in a gene called DLX. The division didn't change the function of this region of the head. The branchial arches still support the gills, just as they always have. However, the changes in the dorsoventral patterning had fairly profound implications for the 2 more interior arches. Next, let's take a look at what the dorsoventral partitioning does to the mandibular arch. The separation of this arch into the upper and lower portions creates a hinge between them, or a natural point of flexion. The lower portion of this structure was already associated with the lower lip, and now becomes the lower portion of a jointed jaw. We also see a major shift in the positioning of the upper portion of the mandibular arch, which now occupies the position formerly occupied by the premandibular region. The premandibular region, in turn, has shrunk and moved a little more dorsally. The premandibular region here has shrunk and moved a little more dorsally, where it seems to have now become incorporated into the ventral surface of the neurocranium in modern fishes. So we have two major changes here. One is the division of the mandibular arch into upper and lower portions, which was caused by the duplication of the DLX gene. There were also changes in the expression of the resulting gene family. The other is a spatial shift in the relationship between the mouth and the mandibular arch. Originally, the mandibular arch formed just the ventral margin of the mouth, with the premandibular region forming the dorsal margin. After the origin of jaws, the mandibular arch forms the entire margin of the mouth, and the premandibular region has receded. These structural shifts create a big functional difference, in that the modified one has the capacity to bite things. In the original morphology, it does not. That huge functional shift was made possible by shifting the developmental program of the head, so that the mandibular arch took on a new position and gained a subdivision that allowed the evolution of the new function. Neat. Now, you may have realized that I haven't mentioned the purple arch yet, which again is called the hyoid arch. Let me go ahead and add it here, so that you can see how it too became subdivided. The hyoid arch also moved anteriorly to become part of the structural support for a newly formed jaw. It also plays an important role in expanding and contracting the oral cavity during ventilation in modern bony fishes, which is the process by which they move water through the head in order to respire. I'm going to talk a lot more about the evolution of this arch in the next in this series of lightboard videos, and we'll show you how a subtle shift in the placement of the upper portion of the hyoid arch catalyzed subsequent diversification of jaw form and function. But for now, I'll just show you what the shift in this arch does to the gill slits. This anterior gill slit has been shifted anterodorsally and has shrunk, but it didn't disappear entirely. We can still see this hole in some modern fishes, most obviously in skates and rays, in which it is a hole in the head called the spiracle and is used during ventilation. Now, to sum up this section, by juxtaposing these two drawings, it becomes abundantly clear that gnathostome jaws originated by modifying the developmental program and the visceral arches of jawless fishes. This is one of the most important events in vertebrate evolution because it gave fishes the ability to bite. Before I let you go, I want to talk briefly about the integration of bone into the jaw. As we've already seen, dermal bone, e.g. the bony head shield, evolves quite early in the history of fishes, and early jawed fishes, as well as some jawed fishes, definitely possessed dermal bone. When we look at the skull of a fearsome Dunkleosteus, like I show on one of the slide sets this week, it's impossible to ignore the bladed lower jaw. However, it is important to realize that these dermal elements are not, strictly speaking, the transformed visceral arches, because the visceral arches are not part of the dermal skeleton. Rather, the original jaw of fishes was cartilaginous, and much of the placoderm jaw was still cartilaginous. The obvious bones associated with the jaws are dermal elements sheathing that cartilage. So let me add some of those elements here to my sketch using the orange color. It's easy to miss this point when we examine placoderm fossils, because bone fossilizes better than cartilage does. That means that in many ancient fish fossils, the entire cartilaginous portion of the skeleton is missing. Not until the evolution of endochondral bone in Osteichthyes do we start to get regular fossilization of the branchiocranium. And with that, I'll close this section of my lightboard lecture. Tune in next time when I talk about differences in how fishes suspend their jaws from the neurocranium and how one particular type, called hyostylic suspension, opened a huge range of additional ecologies to jawed fishes. For today, I hope that you now understand how fishes found their jaws by modifying the development of the cartilaginous branchiocranium, and how that innovation let them put the bite in trilobite.